Greetings to everyone. My name is Dhavan, and today I'll be discussing my project that is based on in-silico analysis of differentially expressed genes and their pathway analysis in prostate cancer. Before moving forward, let me introduce myself. My name is Dhaval Kumar Srivastava, and I'm a final year student pursuing integrated MTech in biotechnology from Amit University, Noida. My research interests include omics data analysis, microbiology, cell and molecular biology, nanobiotechnology, and artificial intelligence and machine learning in healthcare. Starting with prostate cancer. Prostate cancer is the second most common cancer diagnosed in men and the fifth leading cause of death globally. Prostate cancer can be asymptomatic in its early stages and has a dissolute course that may only necessitate active surveillance. Prostate cancer incidence and mortality rates are strongly related to age, with elderly men with age greater than 65 having the highest incidence rates. In comparison to white men, African American men and African men have the highest incidence rates and more aggressive kind of prostate cancer. Early stage prostate cancer can be asymptomatic. However, common complaints including difficulty in urinating, increased frequency, and nocturia, all of which are the signs of prostatic hypertrophy. Because of the axis skeleton in the most prevalent site of bone metastatic illness, more advanced stages of the disease may present with urine incontinence and back pain. Prostate cancer kills an estimated 30,000 men alone in United States each year. Recurrent somatic mutations, copy number alterations, and oncogenic structural DNA rearrangements, such as chromoplexy, have been found in primary prostate cancers in several studies. Point mutations in SPOP, FOXA1, and TP53, as well as copy number changes in MYCRB1, P10 and CHD1 and E26 transformation specific fusions are among the physiological relevant genes. Although several initial prostate cancer changes or signatures have been shown to have prognostic clinical values. Potential tumor biomarkers for early detection and risk assessment of prostate cancers have been identified using comprehensive gene expression profiling. LaPointe et al. In 2004 used a cDNA microarray based analysis to stratify tumors into clinically meaningful subgroups of prostate cancer, which were then linked with tumor grade, stage, and preoperative prostate specific antigens, that is PSA levels. PC81 was found to be involved in prostate cancer progression after transcriptome profiling sequencing was performed on a prostate cancer cohort. Significant intratumoral heterogeneity and a lack of understanding of the molecular mechanisms regulating tumor growth are barriers to efficient clinical management of prostate cancer. As a result, there has been a surge in interest in learning more about prostate cancer pathophysiology during local and distant tumor progression in order to improve diagnostic sensitivity and therapeutic success in the clinic. Transcriptomics, genomics, and other omic technologies have enabled this simultaneous determination of thousands of molecular characteristics, leading to an improved understanding of molecular pathophysiology in chronic heterogeneous diseases such as these. As a result, targeted therapeutic techniques have been developed based on clinically relevant actionable mutant genes, disease-altered pathways, and molecular patterns with prognostic and predictive importance. Molecularly driven therapeutic targets are predicted to improve intervention in advanced prostate cancer as a part of personalized therapy regimens based on innovative and more targeted guidance guided by omics-based biomarkers. So the main objective set for this study is to understand the molecular complexity of prostate cancer by in-silico transcriptomics data analysis. For this, we have obtained a dataset from TCGA-GDC portal via RScript, followed by its normalization, followed by quantile filtering, sample distribution into tumor and normal species, differential expression analysis, enrichment analysis, followed by a pathway analysis. Beginning with, the project was started with importing the TCGA-BioLinks library, followed by retrieving prostate cancer dataset. The project name given to the dataset is PRAD. So a query was put up on RStudio stating the project title as TCGA PRAD under the category of gene expression with a quantified data type, wherein the quantification has been performed on Illumina-seq high-sec with RNA-seq data strategy, ultimately leading to a normalized result. Once we received the output, we downloaded all the 550 files present in it, followed by preparing an expression matrix with gene ID in the rows and samples barcode in the columns and rsym.genes as result as values. After the summarized experiment library was summoned to summarize the experiment, that is to incorporate metadata into the already obtained data, including the clinical data as well. After the incorporation of metadata, a correlation box plot was obtained to check the outliers that could be run, followed by data normalization and quantile filtration with a bar of 0.25, that is the quantile cut of 0.25, from which normal samples, that is the NT samples, and tumor sample as the TT samples were obtained to perform differential expression analysis. Upregulated differentially expressed genes were obtained from normalized filtered data by incorporating a false discovery rate, that is the FDR rate, cutoff at 0.001 and a log fold change threshold at plus 1.5, whereas for downregulated expression uh, analysis of genes for downregulated genes, the false discovery rate, that is the FDR rate, was cut off at 0.001 and a log fold change threshold at minus 0.1. After the DEA, we performed a gene ontology enrichment analysis to identify classes of genes that have been overexpressed and might have an association with the disease. Once we obtained the upregulated and downregulated genes with their log fold change threshold values, we moved ahead with the pathway analysis, which incorporated annotation ontology results from EnrichR, X2Web, and GeneMANIA. The results obtained were as follows. We started the pre-processing phase by building an array-array intensity correlation plot. The gene expression matrix, which has rows representing genes and columns representing samples. Each entry in the matrix represents the expression level of a specific gene. To identify a problematic array, we created an AAIC plot that defines Spearman's symmetric squared correlation matrix between the samples. The color indicates the strength of the correlation between samples. Higher correlations are displayed in darker colors and lower correlations are displayed in lighter colors. Samples considered outliers are removed according to this matrix. That is the second part of the figure, that is 1b. A correlation cutoff of 0.25 was used to determine outliers, and then a normalized process was applied to the prior gene expression data to correctly infer expression levels and avoid errors in the expression measurements. We then moved on to performing differential expression analysis, wherein we obtained a total of 695 differentially expressed genes, out of which 265 were upregulated differentially expressed genes and 430 were downregulated differentially expressed genes. To reduce the underlying biological processes from the DEGs, we performed an enrichment analysis using the TCGA-analyze enrichment analysis component function. The TCGA-BioLink output showed a bar chart with the number of genes assigned to the main categories, such as biological processes, cellular components, molecular functions, and pathways. The top 20 are shown here for each of them. We then combined the differential expression data into a single file and loaded it onto iLinks for further analysis. iLinks is an integrative web-based platform for analysis of omics data and signatures of cellular perturbations. The platform facilitates analysis of user-submitted omics signatures of diseases and cellular components in context of large compendium of pre-computed signatures of more than 200,000, as well as mining and reanalysis of large collection of omics datasets of more than 12,000 pre-computed signatures and their connections. iLink's workflow integrates vast omics data resources and a range of analytics and interactive visualization tools into a comprehensive platform for analysis of omics signatures. We uploaded the gene name and its logFC onto this interface and proceeded to EnrichR, X2K and GeneMANIA analysis. EnrichR is a platform that uniquely integrates knowledge from many high-profile projects to provide synthesized information about mammalian genes and gene sets. The platform provides various methods to compute gene set enrichment, and the results are visualized in several interactive ways. Results from the EnrichR showed that PPAR signaling pathway and AMPK signaling pathway were most significantly expressed, with PPAR leading the pair with a p-value of 8.64 e-8 with an adjusted p-value of 0.000009117, and AMPK stuck with the p-value of 1.558 e-7 with the adjusted p-value similar to the PPAR pathway. PPAR, that is the peroxisome proliferator activated receptors, are nuclear hormone receptors that are activated by fatty acids and their derivatives. PPAR has three isotypes, that is PPAR alpha, PPAR beta and PPAR gamma, showing different expression patterns in vertebrates. PPAR alpha plays an important role in clearance of circulating or cellular lipids via regulation of gene expression involved in lipid metabolism in liver and skeletal muscles. PPAR beta delta is involved in lipid oxidation and cellular proliferation, and PPAR gamma promotes adipocyte differentiation to enhance blood glucose uptake. The AMP activated protein kinase is a serine threonine kinase that is highly conserved through evolution. AMPK system acts as a sensor for cellular energy status. It is activated by the increase in the cellular AMP. AMP-ATP ratio caused by metabolic stress that either interferes with ATP production or the acceleration of ATP consumption. Several upstream kinases include liver kinase B1, LKB1, calcium carbomodulin kinase beta, that is CAMKK beta, and TGF beta activated kinase 1, that is TAK1, can activate AMPK by phosphorylating a threonine residue on its catalytic alpha subunit. Once activated, the AMPK leads to a concomitant inhibition of energy-consuming biosynthetic pathways such as protein, fatty acid, and glucose glycogen synthesis, and activation of ATP-producing catabolic pathways such as fatty acid oxidation and glycolysis. In the activation of AMPK signaling pathway, AMPK can be activated by upstream AMPK kinase, and their sites of action are phosphorylated by AMPK alpha subunit of 172 sulfide amino acid. EnrichR also delivered the data for most significantly differentially expressed genes associated with most common differentially expressed pathways, that is PPAR and AMPK signaling pathways. It was identified that 6 genes, that is ADIPOQ, CD36, PCK1, FABP4, AQP7 and PLIN1 were the significant genes in PPAR signaling pathway, and 8 genes, that is ADIPAQ, CD36, LEP, SLC2A4, PCK1, PFKB1, GYS2 and LIPE were the significant genes in AMPK signaling pathway. After EnrichR, we moved on to X2K web platform for transcription factor enrichment analysis, that is TFEA, kinase enrichment analysis, that is KEA, and to develop an expression to kinase, that is X2K network, on the X2K web platform. The X2K web infers upstream regulatory networks from signatures of differentially expressed genes. By combining transcription factor enrichment analysis, protein-protein interaction network expansion with kinase enrichment analysis, the X2K web produces infer networks of transcription factors, proteins, and kinases predicted to regulate the expansion of the input gene list. The transcription factor enrichment analysis, that is the TFEA, predicts transcription factors that are predicted to regulate the input gene list by performing gene set enrichment analysis using different transcription factor gene set libraries. For example, integrated target genes for transcription factors are determined by ChIP-seq experiments. The results obtained here are then used as an input for the next step involved in the X2K pipeline. For our cohort, the TFEA predicted that PPARG was the transcription factor with most significant association, as it scored 8.01 minus 9, which is the highest value in the negative scale of minor logs 10. The encoded protein PPARG is a ligand activated transcription factor that is involved in the regulation of adipocyte differentiation and glucose homeostasis. The protein forms a heterodimer with retinoid X receptors and binds to DNA motifs termed as peroxisome proliferator response elements to either activate or inhibit the gene expression. TFEA also deduced that PPARG target 9 genes from the input list, that is AQP7, LIPE, CIDEC, CD36, GOS2, FABP4, GPD1, ATP1A2 and LEP. We then moved on to kinase enrichment analysis, which predicted the protein kinases that are likely to be the regulators of the expanded protein-protein interaction network from the previous step. The KEA performed enrichment analysis on the list of proteins from this subnetwork using gene set libraries from kinase substrate interaction databases. For our cohort, the KEA predicted that MAPK1 and MAPK2 were the two proteins kinases that are most significant, since they scored 3.606 minus 16 and 3.736 minus 15 respectively, which were the highest value in the negative scale of log 10. The two MAP kinases that play a key role in the MAPK ERK cascade are MAPK1 ERK2 and MAPK3 ERK1. They also take part in the signaling cascade that is triggered by the activation of KIT and KITL-GSFC. The MAP kinase cascade regulates transcription, translation, and cytoskeletal rearrangements to mediate a variety of biological processes such as cell growth, adhesion, survival, differentiation, depending upon the cellular context. TFEA and KEA was then followed by the expression-to kinase network, which displays the inferred upstream regulatory network predicted to regulate the input list of genes by integrating the results from TFEA and the kinase enrichment. The pink nodes in the diagram of the network represents the top transcription factors predicted to regulate the expression of input gene list. The orange nodes represent proteins that physically interact with the enriched transcription factors and correct them. Blue nodes represent the top predicted protein kinase known to phosphorylate proteins within the expanded subnetwork. The green network shows the edges or links, which represent kinase substrate phosphorylation interaction between kinases and their substrates, while the gray network edges represent the physical protein-protein interactions. Since we received the differentially expressed genes from the differentially expressed pathways, PPAR, AMPK signaling pathway, and the differentially expressed transcription factor, that is PPARG, we found that there were a few differentially expressed genes common between the above three. FABP4 and AQP7 were found to be common between differentially expressed PPAR signaling pathway and differentially expressed transcription factor PPARG. LEP and LIPE were found to be common between differentially expressed AMPK signaling pathway and differentially expressed factor PPARG. ADIPOQ and CD36 were found to be common for differentially expressed PPAR and AMPK signaling pathway, and CD36 was the only differentially expressed gene found common between the above three. Not only that, it was also found that these genes were all downregulated differentially expressed genes. We then moved on to GeneMANIA to predict the function of the differentially expressed CD36 gene found common among all the above mentioned statutes. GeneMANIA finds other genes that are related to a set of input genes using a very large set of functional association data. Association data includes protein and genetic interactions, pathways, co-expression, co-localization, and protein domain stability. The network produced involved 83.50% of co-expression, denoted in lilac, 6.41% of physical interactions, denoted in pink, 3.58% of co-localization, denoted in purple, 2.72% of predicted values, denoted in orange, 2.48% of pathways, denoted in blue, 0.96% of shared protein domains, denoted in yellow, and 0.43% of genetic interactions, denoted in green. From the above experiment, we concluded that the set of genes that were found to be differentially expressed, namely FABP4, AQP7, LEP, LIPE, ADIPOQ, and CD36. CD36 gene holds the potential to serve as a diagnostic test for the purpose of early detection of prostate cancer. In addition, PPARG transcription factor in association with MAP kinase 1 and MAP kinase 3 can also be looked to as a potential proteomic biomarkers to facilitate in the diagnosis of prostate cancer. However, further analysis needs to be performed on these genes to understand if they can be used in fusion with some other genes to increase their selectivity and specificity. Thank you.